Hello, my name is Shayan Sharif. I'm the Associate Dean Research and Graduate Studies for the Ontario Veterinary College. It is indeed my great honor and privilege to introduce to you this year's Chappel Lecture, Dr. Michael Surette. But before doing that, I would like to provide you with some information about the Chappel Lecture. The Chappel Memorial Lecture was established in 1988 through an endowment by Dr. and Mrs. Clifford I. Chappel in memory of Dr. Chappel's father, Herbert James Chappel. It is the intention of those administering the series to bring each year to the University of Guelph campus. And unfortunately, this year we have to do this virtually, a scientist of international repute to address interested members of the student body and faculty. Through such an encounter and through further informal discussions, the university hopes to provide its members with an opportunity to hear people on the forefront in their own research sphere, who will not only summarize their current work, but trace its development and progression over the years. The Chappel Memorial Lecture is intended to be not so much a delineation of a field of study, but as a source of inspiration for those engaged in or considering graduate studies in biomedical research. So it is indeed my great pleasure to introduce to you Dr. Michael Surette, who is without a doubt one of the top microbiologists in Canada and perhaps around the world. Many of you have interacted with Dr. Surette over the years, but for those of you who haven't had the opportunity to know him and interact with him, Dr. Surette completed his PhD at the University of Western Ontario in '91, and after conducting his postdoctoral research at Princeton University, he joined the faculty at the University of Calgary in '98 in the Department of Microbiology and Infectious Disease. In 2010, he moved to McMaster University, and he's currently a professor and a Canada Research Chair in Interdisciplinary Microbiome Research in the Department of Medicine, as well as the Department of Biochemistry and Biomedical Sciences. He's also director of the Farncombe Genomics Facility, and in addition, he's a fellow of the American Academy of Microbiology. Dr. Surette's research addresses the human microbiome of the respiratory and gastrointestinal tract in health and disease, and the lab is focused on developing high-throughput culturing and phenotyping methods to investigate infectious disease and the microbiome, applying and improving next-generation sequencing approaches to characterize the microbiome and exploiting secondary metabolic products of the human microbiome and how the microbiome changes with age. I'd like to turn this over to Dr. Surette.
First, I'd like to certainly thank you for the honor of being able to give the Chappel Lecture this year. It's quite a privilege, and I must say, building up stronger connections with the University of Guelph, so it's extra special. And I wish I could be there and do this in person, but I'm going to do my best to do this. So today I want to talk about animal associated microbiomes in health and disease. Really going to talk mostly about human data, but I will mention some of the work we're involved in. And the last thing I'll talk about, it really does relate to animal health. And I think the lessons that we learn from either animals or humans are interchangeable, and it's a great opportunity for interaction and collaborations. And the microbiome, I think, brings us all together, which is a great topic. So just to give you a little bit of background, again, from a very human perspective, when we talk about the microbiome, of course, we talk about all the microorganisms that live on or in us. And although this was an emerging field 15 years ago, it's hard to avoid the microbiome, and it's even made its way into advertising and just about everything. Every body site on the human body has a distinct microbiome, and one of the remarkable and challenging things about the microbiome is that we each have our very own unique microbiome, which I'll talk about a bit more in a few minutes. We are born essentially sterile. This has been a somewhat of a controversial topic, but a very nice study with colleagues, the Sloboda lab led by Kate Kennedy, I think really established that healthy infants are born sterile, rapidly colonized at birth. It takes about a year for the microbiomes to stabilize. And during this developing microbiome, our immune system is co-evolving, and this really unique interaction between the microbiome and the immune system and each individual helps stabilize this individual microbiome and helps stabilize this community long term over the course of life. And really, as we go through life in the absence of significant disease, the microbiome is pretty stable, even with changing diets and so on. The microbiome remains quite stable throughout life. And then as we age, this microbiome immune system starts to disentangle, and we see large changes in the microbiome, and it certainly loses stability. Of course, what happens very early in life is important, and anything that happens in early life can really affect this trajectory across the lifespan, and that alters our ability, that alters our susceptibility to all sorts of diseases. And so early life events are critical in the development of the microbiome, and perturbations, antibiotics, infections, diet, mode of birth, being breastfed or not breastfed, can alter the risk of developing diseases throughout life. And part of that is how the microbiome and the immune system really established together during that first year, and perturbing that really has long-term consequences. Our lab is really interested in many aspects of the microbiome across the life course. We're involved in a number of studies in early life and prenatal life with the Sloboda lab, and as part of the child study, most recently on a Canadian Microbiome Initiative 2 grant led by PJ Subarero out of University of Toronto. We're also interested in disease, particularly pediatric inflammatory bowel disease. Another CMI2 grant. The Spore Imagine Network is a large cohort of mostly adult, but some pediatric individuals with GI disease. 17 centers across Canada, a few thousand microbiome samples that we're starting to analyze, including lots of healthy controls, studies on type 2 diabetes, with the McNeil and Scherzer lab. And here, I won't talk about this, by using C. elegans as a model to study human microbes and their impact on metabolism. And an area that we're most interested in, or a big program in, is in aging and how the microbiome changes with age, particularly in the respiratory tract, and how that alters susceptibility to disease. So while the primary focus of the lab is on the human microbiome, we do a lot of microbiome work for a number of groups, and we've looked at a lot of organisms. Chickens, chimps, even things like we've done a few sea slugs, spiders, stink bugs, mussels. And what's quite remarkable is many of the microbiomes are somewhat similar. And I'm just going to leave this as a food for thought, because I don't really understand why. But the two outliers here are mouse and rat, and these are laboratory animals, not wild animals. And whether that has something to do with this, I'm not sure. But it was quite striking when we saw this. But we do a lot of work outside of the human microbiome. A few years ago, a young man from Guelph, that many of you may know, came to the lab to spend some time culturing the horse microbiome, and that started a great collaboration with Memo, which has been both an enjoyable friendship and a stimulating research collaboration. And although I'm going to talk about mostly the human microbiome, one of the things I did learn from Memo was, in fact, how complex the microbiomes of larger animals and ruminants are. And everything I'm going to tell you about the complexity and diversity of the microbiome, you can multiply many times over if you're a large animal, if your interest is large animals. And I've also had some very stimulating talks over the years with Cheyenne and learned a lot about poultry production and some interesting possibilities to do some microbiome work with there with Cheyenne, which I hope we'll start to do in the future. So one other thing that we've done, which has also been kind of intriguing, I'm getting more and more interested in, is the role of microbiomes in wild animals, potentially as environmental monitors. This was a study we did with Joanna Wilson and Karen Kidd, two biologists at McMaster, studying fish along the Grand River. And this has been studied for a long time, where you take fish in the more pristine watershed and then sample them downstream of water treatment plants in Kitchener and Waterloo. And it's been known for a long time that these animals are good monitors of long-term environmental exposures, and you can see physiological changes and changes in health of the animals over time. What's interesting is the microbiome may be a better way to look at acute exposures and allow us to look at short-term exposures as things move through the water system. And this is an area which I think is going to be quite interesting. And more recent unpublished data we've been doing with Karen Kidd has been looking at aquatic invertebrates and riparian spiders along the same water system. And there's an impact on the microbiome of these invertebrates. But one of the things that really struck out to me, which I think is going to be interesting to follow as we move further, is that we do see a significant impact on the endosymbionts in some of these organisms. So endosymbionts are bacteria that actually live within the cells of the host cells and are passed generation to generation, and have a huge impact on the physiology and the reproductive biology of the host animal, and are sort of independent of environmental contamination with other microbes, because they're kind of unique. And we see a pretty significant change. And so this is an area I'm quite interested in. And one of the impacts of COVID was I lost the opportunity. Last summer, I was supposed to go collect spiders with the biologist, which I was looking forward to. But hopefully when COVID is over, I can do some field biology, which is something my lab doesn't really do very often. But again, I think this is an aspect of the microbiome I hadn't really thought much about before, but I think has lots of potential on the environmental side. But I'm going to tell you mostly about the human microbiome, and we know that the human gut microbiome is really integral to almost all aspects of human health and disease. And for 15 years now, we've seen evidence to show that changes in the gut microbiome are correlated with a wide range of disease. Clearly gut diseases, but diseases like asthma, cardiovascular disease, and even psychiatric disease, anxiety and depression, and growing evidence for things like dementia and so on. But one of the challenges has been really hard to get at mechanism. So I'm just going to take a couple of slides to explain why this is such a hard problem, and then I'll get on to more positive things about the microbiome. But there's really three main things that make this difficult. And so I just want to highlight those. And the first is that everybody has their own unique microbiome. And this plot is showing a PCOA plot, which is a measure of looking at the difference between microbiomes. So each of the dots here represents a different individual, different microbiome. The distance between the spots really represents how different they are. So the closer together they are, the more similar. And this is healthy controls in ulcerative colitis patients, and this is looking at the stool microbiome. Healthy controls in red, and the patients in blue, and you can see that there's lots of overlap. There's a little bit of separation, and if you took the mean values of the healthy controls and the mean values of the ulcerative colitis, they would be different. But at an individual level, you really can't tell. And if you look at the plots without the colors, you really can't pick out who has ulcerative colitis and who is a healthy control. So this intrinsic diversity in the microbiome is really a challenge, and we can't look at a microbiome and say, aha, that's a healthy microbiome, and this is a diseased microbiome. So that's actually a huge challenge. In some agricultural animals, the microbiomes may be much more similar between animals, and that might be less of a problem. But in general, this individual heterogeneity is great in other aspects, but challenging in trying to dissect some of the roles of the microbiome. Another problem is there's a lot of functional redundancy. Butyrate is a good example. Butyrate is a short-chain fatty acid produced by the microbiome. It's really integral to GI health. It's an important molecule within the gastrointestinal tract. And if you're low in butyrate, the GI tract becomes leaky and permeable, leading to systemic inflammation. Butyrate also modulates epigenetic modifications in the GI tract. It modulates hematopoiesis in the bone marrow, spleen cells. It can reach the lungs and alter immune properties in the lungs, and growing evidence suggests that it can get to the brain and affect brain function. There are 4 different pathways for butyrate synthesis in microbes, and there are many different bacteria that are capable of doing it. This is just a short list of a few genera that are butyrate producers. Within each of these genera, there's multiple species, some which do and some which don't produce butyrate. And then within a particular species, some strains may be better than other strains at producing butyrate. So just knowing you have one of these strains doesn't always tell you that you're capable of producing butyrate, and you're missing one of these organisms. There's another organism that can replace it. So this redundancy makes the comparative analysis difficult in trying to understand what's present and what's absent. The other thing is the opposite of redundancy, and that is real strain specificity, and where a property might be a function may be a property of a strain, but not a whole species. And there's a really good example of this from Peter Turnbau's lab from a number of years ago, but it's such a great example. Digoxin is a common medication for heart conditions, and it's used quite widely. It's been known for a long time that there are patients that just don't respond to that. And the non-responders excrete a modified version of digoxin, which has been modified and secreted and doesn't accumulate in the blood and is not active. And it's also been known for quite some time, since the 80s, that if you add broad spectrum antibiotics to the patients, or before giving the drug, the digoxin actually stays in circulation and doesn't get modified, which really suggests that there's a microbial component to the degradation. And also, digging through the literature, they found that actually a member of the human gut microbiome, the Ergothella, is able to carry out this reaction in vitro. So what they did was they got Ergothella from a strain bank, showed that these 2 genes were in fact responsible for putting on this modification, and in all of the non-responders, they were able to go in by PCR and show that these 2 genes were present, and they weren't present in the responders. So this organism and these genes are really responsible for this. But the actual story is, it was only the 3rd isolate that they got from the strain collection that was capable of activating digoxin and had these enzymes. So it's not a matter of having this organism, it's a matter of having the right strain of this organism which has these genes. And so this strain specificity and these strain specific functions adds another layer of complexity to the microbiome, which makes it, again, a challenge to study. So what I'd like to do, again, is for the rest of my talk, is to talk about the diversity of the host microbiome, which is remarkable, untapped resource of science that has been fascinating to get involved with, and is going to keep us busy for a long time. And I'm going to briefly talk about one of the strategies that we use, which is culture and culture-enriched approaches, and then give 3 short vignettes on some ongoing research. So it was widely held belief for a long time, and I think this is slowly changing, that culture really just shows you a small tip of the iceberg of the microbiome, and that culture-independent methods show you the whole iceberg, all the stuff that you can't see by culturing. And this is widely assumed across microbes in the environment as well, but has been assumed to be true of the host-associated microbes as well. And certainly we don't believe that in my lab. We're very focused on culture. And I hope to convince you in a few slides that, in fact, culture-independent methods show you the tip of the iceberg, and culture methods, particularly combined with deep sequencing, really show us the true diversity. And of course, this shouldn't be a surprise to this audience. Emma Allen Burko in biological sciences is one of the great cultures of the microbiome, and I'm sure many of you are familiar with her work. She was many of the early believers that the microbiome was indeed culturable, and has done great work along those lines. So what we do is not particularly complicated. So if you start with a sample, we can ask what bacteria are present using 16S profiling. This is where we sequence a small sequence of the ribosomal genes. We can identify what those are, and that tells us essentially what's present in the sample. And this is basically bacterial profiling, and has been the way that a lot of the microbiome research has been done for the last decade or so. What we do is we plate under a large number of conditions, both selective and non-selective media, aerobic conditions, anaerobic conditions, sometimes microaerophilic conditions. And then we treat each of these plates as a different community, and we sequence the 16S, the plates with 16S, and see, what bacteria have we grown, and we can compare what's present here to what we grew, and ask, can we culture most of what's there? And we did this in the airways back in 2011, and 2016, Jen Lau, a graduate student in my lab, published the protocol really for the airways. We've refined that a bit, but Jen's work originally worked with 66 growth conditions. It was quite a bit. We've trimmed that somewhat, but it does allow you to culture lots of things if you use lots of conditions. And sure enough, we could culture the vast majority of the microbiome, the human gut microbiome under these conditions. But what was more striking, so there's about 120 bacteria that were identified by doing 16S sequencing on the stool on this individual. We've done this on many individuals. But we asked how many of those could we get when we cultured, and there's one or two that we didn't get, I think two. But when we asked, was there anything we cultured that we didn't see when we just did this sequencing on the direct sample, we cultured about 4 or 5 times more organisms. And typically, when we do the culturing, we see 3 to 5 times more organisms than we detect even by deep sequencing. And this is really true of other groups that have done similar work, that culturing really reveals diversity that we don't see. And this is because culture selects for low abundant organisms. It reduces the growth under particular conditions of things that might grow fast. And so you just capture more diversity using this approach. And I will emphasize that this is 16-based profiling, so it's really at best at the species level. It doesn't talk at all about the strain diversity, which I mentioned a minute ago. But this really does allow us to get at the microbiome, depth of the microbiome much better. And more recently, we've gone from doing 16S sequencing to shotgun metagenomics. And this is where, instead of just amplifying and sequencing a single gene to get an idea of taxonomically what's there, the idea is you sequence all the DNA and you should know all the genes that are there, and you can try and put together genomes. So when I refer to direct metagenomics, or you see DMG, it's basically isolating the DNA from the sample and doing shotgun sequencing and trying to put it all together. Using the same strategy, what we can do is we can do shotgun metagenomics on the different plates, and that's what we call culture-enriched metagenomics. Culture-enriched metagenomics significantly increases the length of contig assembly. It allows us to assemble large fragments of genomes. It allows us to put genomes together from the sequencing data significantly more. And it really increases the number of functional genes and pathways in any category that we're able to identify. So it gives us a much deeper look into the microbiome. Just to show you a bit of data, this is samples from a donor that we've used in a number of fecal transplant studies to study ulcerative colitis, which we won't talk about today, but we've done culture-enriched metagenomics on this individual. This is the best case scenario. We have 4 direct metagenomic samples over 5 years for this individual. So this is as good as direct metagenomic gets. And when we combine all that together, we get about 300 million base pairs of assembled microbial DNA greater than 2.5 kb. So these are pieces of DNA large enough to hold a couple of genes, not just the small fragments, which we ignore. We get about 49 assembled genomes and about 110 bins that contain partially assembled genomes or mixed genomes, and a bunch of sequences that don't fit into anything, which are very interesting, of course, but about 300 million base pairs of assembled sequence. The culture enriched data from this gives us about 700 million base pairs of assembled data, 255 assembled genomes, 272 bins, and much higher resolution of the genome of the microbiome that we can see, really with lots of direct metagenomics. And this allows us to really study this in great detail. And in the context of fecal transplants, we've been able to show strain level transfer in a few instances between donor and patient. And just to put the amount of information here in context, the human exome, which is the coding part of the human genome, is about 30 million base pairs. And bacterial genomes are 90 to 95% coding. So there's a lot of genes and information in here which far outstrip the human gene content when you think about the gut microbiome. For any aficionados in the field, this is a bit specific to people who really think about this stuff, but when we assemble megs using direct metagenomics, they're always smaller than the megs that we assemble with culture-enriched metagenomics, and we really actually start to pull apart assembly of different strains in the database with the culture-enriched metagenomics. So it actually gives us better megs than we can get from the direct metagenomics. So I'm going to tell you a couple of stories about looking at the diversity. And the first one I'm going to talk about has to do with sugar. And microbes live really in a sugar-coated world. And to paraphrase Morpheus, the glycome is everywhere. All cells, microbial and host, are decorated with carbohydrates. And these play essential roles in the interaction between host and microbes. The human host is covered in a variety of sugars on the surface, and extracellular matrix is decorated in sugars, and these complex polysaccharides really form the interface of the microbiome and the host. And the enzymes that play around with these sugars, glycosyl hydrolases, cleave these hydrolytic bonds. And we're very interested and have started to work on glycosyl hydrolases. And when we look into our culture-enriched metagenomes, this is 6 different culture-enriched metagenomes, we have on average about 10,000 glycosyl hydrolase genes in each of these metagenomes. And that example I showed you before, which is the one where we've actually sequenced the deepest, we actually have 14,000 glycosyl hydrolases really from a single sample of stool. So this is a rich diversity of enzymes that plays an important role with an area of host microbe interaction that really doesn't get enough attention. This story has really been driven by Evan Mann, and this is a Guelph story. Evan is a terrific biochemist and burgeoning biochemist or a bioinformatician in this area. And Evan was a PhD student with Chris Whitfield and joined my lab as a postdoc a couple of years ago and really initiated this program. Let's talk a little bit about these glycosyl hydrolases. These cleave very specific bonds between 2 sugars and break them apart. So that's essentially what glycosyl hydrolases do. There's a variety of sugars. These are the most common sugars that the humans produce, and these form complex sugars, but there's lots of others. Sugars have specific positions and different types of bonds that connect them, and each of these glycosyl hydrolases families is specific for a very specific configuration of the bond and linkages. This is actually a very well studied and well defined a group of enzymes. They're divided into families. Each of the families is actually a distinct structural fold. And so here you can see the different structures for some of these glycosyl hydrolases. And each of these families cleaves a very specific bond. And so the GH33 is one that cleaves sialic acid or neuraminidase linkages, which is a very common linkage in the human microbiome. I'm going to tell you a story about sialases in a second. So it cleaves a very specific bond to release the sialic acid in that case. But the actual substrate, where that sialic acid is, whether it's on the surface of a protein or a complex polycarbohydrate, is different. So while the enzymology is very specific, the actual macromolecular substrate can be very different. And so there's a lot we don't know about these enzymes. And the number of enzymes and families grow exponentially as we sequence more, but actually the number of functionally characterized ones is pretty small. And so there's a lot we don't know about this family. And it is a huge component of the microbiome. And we're learning more. The number of glycosyl hydrolase families is continuing to grow at a linear rate. And in the context of the human microbiome, we have lots of what are called oglycan linkages, and a good example of that is mucin. Mucin is covered in these glycan groups, and the mucin performs an important interaction between the microbes and the host, and acts as a barrier to keep most of the microbes away from the epithelium. And in lots of GI disease, we see a breakdown of the mucus and allows the bacteria to really contact the epithelium. But another important thing that we often don't think about or haven't thought enough about is that actually the surface of the cells are covered with N-glycans, and these N-link glycans sit on the surface of every receptor. Every protein in the cell is modified in this way. A good example of this is, for example, ACE2, the receptor of SARS-CoV-2, which is doesn't need an explanation, but this glycosylation on ACE2 is critical for the interaction between SARS-CoV-2 spike protein and ACE2 and for internalization. So this glycosylation really modulates function. And we're very interested to know how microbial glycosyl hydrolases may modulate surface receptors and modify the interaction of the microbiome and the host. And this is a new area that's emerging in the lab and one we're very excited about. So about the time that Evan joined the lab a couple of years ago, there was a new family of sialases described. This was glycosyl hydrolase family 156. There was one member. It was found from a hot spring shotgun cloning library and screening. They screened a library of DNA from a hot spring and found an enzyme that cleaves sialase, solved the structure. It's a novel structure, a new family. Previously, only glycosyl hydrolase 33 family was known to cleave these. And so the question that Evan set out was, there was one of these described. Are there any of these in fact encoded in the human microbiome? Evan, as a biochemist, looked at the active site, used the active site and the structure of this to develop a hidden Markov model to look for this class of enzymes, screen all the databases of sequenced bacterial genomes, sequenced and publicly available metagenomes. It's a huge data set, and found a large number of these were associated with environmental organisms. But we were seeing organisms associated with the human microbiome, particularly the Verochrome microbiota and the Bacteroides and the Firmicutes. And then he went in and looked a little closer and did sequence similarity networks. And these are all members of this, predicted members of this glycosyl hydrolase family. This one labeled in green is the only one that's been characterized. All the purple ones are examples of organisms that are associated with the human microbiome, and anyone that has a dark circle here is in our strain collection. And so Evan's gone in and started to clone these enzymes and start to look for activity and demonstrated indeed these are glycosyl hydrolases. Some very important members of the human microbiome have these activities. The next step is to understand if they work on N-glycans or oglycans and start to tease apart the different functionalities. But this is a new direction for the lab, and we think these are going to turn out to be important for a large number of microbe-host interactions, not just in the gut, but at other body sites. And Evan has really initiated this and has been joined by grad student, Claudia Esteban, and Kennedy Austin's a co-op student who formed the glycosyl hydrolase group. This is one of the areas where this amazing diversity of the microbiome has lots of room for exploration. To switch gears a little bit, you can't really work at McMaster and not think about antibiotic resistance genes, because AMR is a big thing at Mac. And so what I want to show you is going back to that first microbiome that I showed you and ask about antibiotic resistance genes. So if we look in the direct metagenomic sequencing of that donor sample, single stool sample, it's hard to see here, but these represent about 115 little blue dots. Those are all antimicrobial resistance genes that are detected. So there's about 115 that we detect by direct culture and or direct sequencing of the sample. This is a healthy individual that had not had antibiotics for years. So this is intrinsic microbiome resistance, or intrinsic antibiotic resistance within the microbiome. Now, to go back to the culturing, the question is, well, how much of the resistome did we culture? And so each of these dots now here represents a different culture condition, and all of the antibiotic resistance genes that were captured on the different plates. And basically, we captured all of these. We didn't get these 8 antimicrobial resistance genes, and it may have been that if we sequenced more plates, we might have had it. Or maybe these are organisms that we just didn't capture. But you can see that we capture the vast majority of the organism or the resistance genes that were present in the direct metagenomics by culturing. And again, we can ask the question, well, are there antibiotic resistance genes that we didn't see in the direct metagenomics, but we saw in the culture enriched metagenomics? And this is what this is. This is again about 4 times more genes than we see in the direct metagenomics, a lot more diversity. And again, this just shows that in the healthy microbiome, lots of antimicrobial resistance genes are floating around in the microbiome. Over the last few years, we've built up a pretty good sequence genome collection. We have about 1,300 gut isolates and about 300 respiratory genomes, for which we have high quality genome assemblies. So not just the isolates, but the assemblies. We have lots more isolates, but these are with really high quality genomes. We developed a nice, inexpensive, long read, short read hybrid sequencing method that really allows for high quality genomes, and that's been important for lots of studies. So thinking about antibiotic resistance genomes or antibiotic resistance, Hooman Durkashani, who I'm going to talk about next, decided to just look at the resistance genes in our collection, and 102 isolates had TET resistance, which is the most common resistance, maybe not too surprising, but interesting. There's a rich abundance of these in the Lactobacillus, which is an important group of gut microbes, which are really among the organisms we think of as the really good gut microbes, obligate anaerobes, and lots of diversity. So Hooman decided to look into this a bit more. He looked specifically in the Lactobacillus. Our collection has 132 isolates covering 22 genera, and these are all the TET resistance genes, or all the resistance genes. The blue ones are the TET resistance genes. They're the most abundant. TET-O resistant was the most abundant resistance gene that he found. But when he started to look just a little bit closer at the data, it turns out that the TET-O sequences were often identical between isolates, even from quite distant genera. And this raises the question about horizontal gene transfer, of course, and so he decided to look into that. And so there are really 3 main mechanisms of horizontal gene transfer. Things can get moved around by bacteriophage, conjugated plasmids, or these integrative conjugative elements called ICE elements, and I'm going to talk about those. These are large mobile, complex mobile elements that are associated with moving around antimicrobial resistance and virulence genes. They have a lot of different components to them, and they're quite large elements, as you'll see in a minute. So what he did was he took the resistance gene, he looked at the flanking regions upstream and downstream of the resistance genes and looked for elements required for ICE elements, so resolvases, pili elements for transfer, relaxases, and so on that are required to make a functional ICE element. And sure enough, he finds lots of examples of those. So these are the resistance genes, and these are the resistance genes that in the flanking regions have components of these ICE elements. So it's quite clear within the Lactobacilli that there are these ICE elements that can move around antibiotic resistance genes, which is something that we really didn't necessarily think of. In one of our highly our closed assemblies, he noticed that there was multiple ICE elements in the same strain for the same thing, and he started to dig a little deeper. And looking around, he noticed that in fact one of these was quite novel. In addition to the TETO, there were a bunch of other genes associated as cargo, which are moved around in the ICE element. So that prompted him to take the ICE element component. So he took 20 base pairs downstream, which contains all of those relaxases, the DNA transferase, pili, and so on, and looked for just this part of the element against all the Lactobacilli genomes. And he found lots more of these ICE elements, but these didn't carry antibiotic resistance genes. They actually carried large clusters of genes, 50 to 60 kb in size, that carried novel functions, and many of these carried pathways for secondary metabolite production, such as non-ribosomal peptides and occasionally polyketide pathways. So this is an example where humans discovered ICE elements, which we usually think of as transferring antimicrobial resistance genes, are now carrying around novel functions that are moving around among the microbiome. And these were quite unexpected. This is just a very quick summary, and if you look across, these are the different organisms. Some might have one ICE element, some have multiple ICE elements. Many of these ICE elements are very common and found in many species, and so there's lots of diversity across the Lactobacilli for these elements. I'm not going to have time to talk about it today, but he was quite interested in looking how widespread these were, and so he looked across the database of all the available Lactobacilli. And what's interesting is he's not been able to find any non-human Lactobacilli that carry these ICE elements. So it seems, at least to this point, that these ICE elements are quite unique to human, and they're not found, for example, in the pig metagenome, for which there's actually quite good data. And surprisingly, the Lactobacilli in pigs don't seem to have these ICE elements. So there's something quite unique, or appears to be quite unique about these. And again, moving around within the genome, or moving around within the microbiome, conferring some advantage to their host, which we're not, we're trying to understand. So I'm going to tell you, finish with another story from Hooman, and to give you a bit of background on Hooman. Hooman was actually grew up on a dairy farm in Iran. He's a trained vet, was a practicing vet for a number of years. Decided he wanted to do veterinary microbiology, did a master's in Australia, a PhD at the University of Manitoba in veterinary microbiology, and decided that he was more interested in the research side of things and decided to pursue research. This timed perfectly with when we were about to start a large culture study of the human microbiome, and managed to distract him for a couple of years to work on the human microbiome, which I just described. But his real passion is veterinary medicine, and he really, mostly on his initiative, has started a program with Cargill looking at bovine mastitis, which is one of his real interests, and trying to understand mastitis. And in particular, what we're interested in is what's the therapeutic potential of the commensal microbiota of the mammary gland and the milk microbiome that might help prevent mastitis and infections. And so he started a program to isolate and characterize commensal microbiomes from healthy mammary glands and test them for antimicrobial against mastitis pathogens. And this is ongoing work with Cargill, and we're in our second round of studying to really look at colonization resistance, which is something we're really interested in. And here, looking specifically at antimicrobial factors in the commensals. The next phase of this will also be to look at immunomodulatory activities of these organisms in this context and see whether they can provide additional benefits. But the strategy has been to collect milk and use this aerobic, anaerobic, and extensive culturing conditions, which we've optimized, and screen for function. So we have a biobank of about 1,500 milk isolates now, 164 unique species, quite a wide distribution of organisms. These have all been screened for antimicrobial activity against multiple mastitis pathogens. And I will again shout out to Guelph. The Guelph Veterinary Microbiology Group was kind enough to provide us with some of the ones that we didn't have. And what I'm showing here are a number of our isolates that have gone on to the next round of screening. These are all mastitis pathogens. If the circle's filled, these have potent antimicrobial activity. So this has antimicrobial activity against all the streps. There's a broad activity against the streps, the staph, and even the E. coli, and so on. And so we're seeing lots of activity of commensals against the microbiome, against the mastitis pathogens. Human's gone on to start to purify some of these extracts. We have really potent extracts, and are slowly working towards trying to identify the molecules involved. We have, of course, whole genome sequence of these. We see lots of bacteriocins, non-ribosomal peptides, polyketides, and other biosynthetic pathways, which may be responsible for making these antimicrobial activities. And for us, exploring the potential of the commensal microbiome to have antimicrobial activity against pathogens is one of the important steps forward, I think, in certainly reducing antibiotic use in agricultural animals. But this synergizes very closely with our work on aging, where the aging microbiome changes and we become more susceptible to infection. And so we're actually doing a similar thing on the human microbiome in the airways and looking for microbes in the respiratory tract that can inhibit colonization and pathogenesis from some common airway pathogens. But this work has really proceeded much faster and has become exciting very quickly, really driven almost entirely by Human, who is an outstanding molecular biologist, microbiologist, has really streamlined and improved the efficiency and reduced the cost of all our genomics and metagenomics. And somewhere in his spare time, he's sequenced a huge number of the SARS-CoV-2 sequences that have been generated in Canada. Our lab's gotten involved in that for a short diversion, and he was really instrumental in that. So he's really pioneered a couple of these big projects, and has really gotten us into the bovine work in a big way. And this is certainly an exciting area of development in the lab. So this has been a pretty quick overview of a bunch of things going on in the lab. Not anything in too much detail, but I hope I've convinced you that we really talk about it, but we do know that the animal associated microbiomes really are important and really affect all aspects of disease. I hope I've convinced you that microbes are, for the most part, readily cultured, and that culturing really gives us so much more information than we get from direct metagenomic sequencing alone. And more important, if you have a microbe, you can really study it in ways you can't study sequence. While I focus today on the gut microbiome, everything is true for the airways and other body sites, and we do very similar things in the airways. Although the bacterial load in the airways is much lower, the bacterial diversity is just as rich. Something I also didn't talk about is infectious disease, which is really how we get into the microbiome. And we certainly at non-sterile sites, like the airways and the gut, have lots of infectious that have unknown etiology and polymicrobial infections, which we don't understand at all. And these same approaches, I think, are really applicable there. And I will comment that frequently my vet colleagues will say, well, you know, we don't have very good microbiology for infectious disease sometimes. And I would say the same thing is true in the human scene. 50% of community-acquired pneumonias are culture negative, which is, to me, quite shocking. So using these same strategies of culture-enriched metagenomics and culturing and really understanding the organisms has lots of potential in infectious disease, and I think an area, lots of application and potential in the One Health arena, and really that's what I'd like to end on. You know, microbes are really at the interface of human, animal, and the environment, and integral in this One Health concept, and we're really interested in seeing the overlap in pathogens, overlap in the potential beneficial roles of the microbiome, not just in humans, but also in animal health, and potentially as monitors of environmental health. And maybe we can do that with spiders, which is kind of fun to think about. And so I hope I managed to put a little something in there for everybody. I'm enthusiastic about some of the things that I talked about, the potential to expand our work in the One Health arena for sure, and some of my valued colleagues at Guelph, which I hope will expand our studies. And with that, I'd just like to thank the lab members and the many granting agencies. I can't put all of the collaborators on this slide, but I'm more than happy to take any question. And thank you once again for nominating me and putting up for this very prestigious lecture. I appreciate it very much.
Mike, that was fantastic. Thanks very much for such an interesting and in a sense mind-boggling presentation, in the sense that, you know, it really opens up, you know, our eyes to how much we know, and at the same time how little we know about the microbiome world, and, you know, how it applies to human and animal health and disease. I'm hoping that we would be able to unshare your screen so that we can have your. No, you can only see me. But anyways, I'm sure that our producer, Kevin Hogg, would be able to toggle between me and yourself. We have question number 1 from Heather Morphy. The question, and I don't actually know if, Mike, you have the ability to look at the chat. So Heather is asking about AMR work that you've done, and she was wondering if there are some risk factors besides antibiotic use that could impact the composition of antimicrobial resistance genes that are found in the gut microbiome of the specimens that you've gathered in your repository.
That's a great question, and I guess I think in the bigger context, ecologically, we really don't know why all these resistance genes are around. Certainly, there's more resistance genes in these commensal gut organisms than we expected. And we do know this individuals had relatively low antibiotic exposure, certainly over the short term. So we were a bit surprised. There wasn't a strong antibiotic selective pressure, at least recently. But these genes are widespread in the environment. They know how to get around. They're on mobile elements. And so perhaps it's not surprising. It's probably not surprising that the TET gene, the tetracycline resistance, was one of the ones that was most common, because that also is one of the most common ones in the environment. But I think we don't know much about these interactions. And the other thing is some of these genes, so some of these genes are fairly specific in what they do, but some of these resistance genes, for example, genes that modify their targets, so glycosylate, adenylate, hydrolase, add hydroxyl groups, and so on. You know, these kinds of drug modifying enzymes may actually act on many other things. And one of the areas we're interested in is this a source of microbial enzyme activity on pharmaceuticals, which we know there's a lot of interaction. So I think we just don't know what all of these genes are doing. And although we tag them as antimicrobial resistance genes, some of these genes might have other functions. And then the microbes are really, you know, we talk about this as a big community, but they really are competing with each other. And they really try to kill each other all the time. So a lot of these are also defense mechanisms against each other. But we don't see typical antibiotics produced in these organisms so often. So I think it's a mystery. We just really don't know enough. And if we could look historically to the pre-antibiotic area into the microbiome and see if these are still there, I think that would tell us a lot about whether this is an artifact of the modern world or whether these have always been there. But I think that's a really good open question.
Very good. Thanks very much, Mike. We have two more questions, and I'm hoping that we would be able to squeeze them in. Question number one is more methodology regarding direct metagenomics and cultured enriched metagenomics. What are your thoughts about the quantity aspect of each method? And as you mentioned, when culturing, it is possible that some bacterial species grow more, and as a result of that, they can dominate other bacteria.
Yeah, so the biggest challenge in culturing the microbiome is to keep the fast-growing things down more than trying to find how things grow. So almost anything we isolate grows on our favorite media. So it's not hard to get things to grow. It's hard to get rare things to grow so you can find them among the weeds. And so that's a big problem. And so often what we're doing is we're trying to find conditions where some of the things we don't want to grow don't grow. That's one of the challenges. The culture enriched metagenomics does lose the quantitative nature, though, and I think that's what you're getting at. So what grows on a plate, not just even in colony numbers, so you can have a big colony and a small colony. When you sequence those, there's very different amounts of DNA that show up, even though quantitatively they actually came from one thing, same number of cells. And so the culture-enriched metagenomics skews the quantitative nature of the data significantly, but we actually can use the direct metagenomics to renormalize that, at least for the abundant organisms. So we do lose quantitative information. To some extent, although we get quantitative information on viable cells and real CFU counts, but you do lose some of the quantitative nature, which you can use the direct metagenomics or even 16S to normalize to some extent. I think that's what you're getting at.
Very good, Mike. And I wish, we had more time, for follow-up questions, but, we are running out of time. We've already run out of time. And I have two more questions, one of which probably requires, more than, a few seconds, but might as well, put it on the table and see how you react to that. And by the way, if there's if there are any further questions, by all means, I just posted the student and postdoc link. You're more than welcome to attend, and Dr. Surette would be able to answer those questions. This question is about the method that you're using, culture sequence and identification of resistance genes, and how that would be, how that methodology could be utilized for antibiotic selection, for example, against bacteria such as Mycoplasma.
So I'm not quite sure I get the question, understand the question. So can we use that to use that information to selectively target specific organisms? I'm not. I want to make sure I answer that question correctly.
Yeah. So the question reads: how do you envision this method of culture sequence and identify resistance genes could allow for better antibiotic selection for infections caused by Mycoplasma?
Yeah, so could we use this essentially as part of antibiotic stewardship in a way? And I think as we're applying it, it's not very applicable. Even in the best case scenario, it's days to generate data this way. There's another strategy to look for antimicrobial resistance genes. There's multiple strategies, but one that we've been working with the Wright lab and the Poinar lab on is using bait capture methodology, where you can go in and capture antimicrobial resistance genes in a sample, and just sequence those. And we're really interested in making that methodology rapid, so it could be a potential diagnostic. So all of the resistance genes in a sample. This has worked quite well, and we benchmarked some of those methods against our deep culture enriched data sets, and you do pull out these genes. So it does work quite well, and it's a little bit more rapid. I still think it's tricky. There's lots of complicated ways which antibiotics can interact with the microbiome and specific organisms, and so I still think it's going to be challenging to sort of use that kind of molecular data as the be all and end all of guiding resistance therapy or treatment therapies. But I do think it's a step forward, and I think in many instances that kind of rapid understanding of the landscape of resistance in a sample is going to be helpful. And I think some of these newer technologies will allow us to do this molecular profiling very rapidly. Happy to talk more about that offline if you're interested.
Very good, Mike. There's one last question, but I'm really conscious of time. It's 1:33, and I see that people are dropping out. And by the way, I should tell you that I was reminiscing about, the pre-pandemic era where we had face-to-face interactions, and I was hoping, to have the same level of turnout today. But I can tell you that the level of turnout today was absolutely fantastic. We had more than 70 people at the peak level, which was halfway through the presentation. So I think, this is even much better than face-to-face interaction in some respect, because, it's quite accessible and reachable for people. This last question is about ICE-mediated horizontal gene transfer, but if I may defer this, to your postdoc and student meeting, I would appreciate it, because I know that people are going to have to move on to their next commitment. So having said that, Mike, again, we are absolutely fortunate to have had you today, and we hope to have more interactions in the future. So thank you again for accepting our invitation to be the 2021 Chappel Lecture. It's been indeed a great honor and pleasure. And I also wanted to thank all the participants, all the attendees, and wish you a fantastic day, rest of the day, and also a fantastic weekend. So take care of yourself, everyone, and have a good day. Bye.
Thanks very much. It's been my pleasure.
Thank you.